benzene, C6H6, a common compound that can be found quite easily in industries such as the petrochemical industry, over here, and plastics industry. Benzene can also be formed from volcanoes and forest fires, leading to high exposure of this toxic chemical to human beings. Ocean spillage and dumping of industrial wastewater exposes the toxins to humans, as well as dumping of industrial waste discharges and sites, causing benzene to contaminate the soil. In USA, more than 100,000 soil and groundwater sites have been found to be contaminated with benzene. However, natural exposure to benzene does not cause benzene poisoning to occur due to the relatively fast rate of chemical degradation in air, water and soil, which ranges from a few hours to a few days maximum. People who work in petrochemical industries or those who smoke tobacco are at the highest risk of being exposed to it, as inhalation of benzene being the most dominant pathway for human exposure. In the body, benzene undergoes many stages of oxidation with cytochrome P450 enzyme that results in the formation of metabolites. The main metabolites that show the most toxic results are trans-muconaldehyde, quinol, and P-benzoquinone. Let's trace the pathway down, starting from benzene. Benzene is first oxidized with the help of cytochrome P450 into benzene oxide, breaking one of the double bonds to form an intermediate benzene oxide. From benzene oxide, it can take a few paths, but let's focus on the one leading to the one benzene diol first. Here, the aromatic ring gets destabilized in order to become more reactive. The destabilization is catalyzed by dioxygenase catalyzed reaction between benzene and molecular oxygen, resulting in the formation of benzene diol. The destabilized ring undergoes a reaction called ring cleavage, where it changes into a trans-muconaldehyde, or called TTM. TTM modifies structure and the plasma membrane of macrophages, causing it to be unable to function as a key cell for the immune system. Besides the formation of benzene diol, the benzene oxide could also be reduced to form phenol. This phenol then undergoes two different pathways of reaction to form two major products. Firstly, the formation of benzoquinone will be discussed. The phenol undergoes a hydration catalyzed by cytochrome P450 to form quinol. The quinol formed will then interact with the oxygen in the body to have the oxygen gain an electron. The oxygen will then attract the proton hydrogen from the quinol to form a hydroxyl radical, leaving the oxygen on quinol with a negative charge. This process continues until both the oxygen in the quinol are left with a negative charge, and a double bond in the benzene ring would be broken to stabilize the negatively charged oxygens to form P-benzoquinone. P-benzoquinone can bind to glutathione, proteins, and such to become a potential bone marrow toxin. This is the result of the opening of the benzene ring, forming a direct alkylating agent which can interact with cellular thiol and various amino groups. The second pathway taken will be to form catechol, or O-hydroxyphenol. With the hydration of the phenol catalyzed by cytochrome P450, this catechol will undergo further hydration to give rise to 1,2,4-trihydroxybenzene. The catechol is the main compound that causes problems within bone marrows by interfering with microtubule assemblies that will result in the arrest of cell division and suppression of lymphocytes pathogenesis. With benzene undergoing such reactions, numerous problems occur within the body. Such problems include anemia, diminished bone marrow function that results in lack of white blood cells, dizziness, and possibly death.